going to be talking about the effects of acute stress on problem solving. So the first study that I looked at was conducted by Duan et al. In 2020. Some background for this study. Acute stress can impair core executive functions such as cognitive flexibility, inhibition, and working memory, which are necessary for problem solving. We know that acute stress affects both the sympathetic nervous system and the HPA axis. So there are two different types of problem solving. There's convergent problem solving, where subjects have a single correct answer to find. And in these, performance is gauged in terms of response accuracy and speed. There's also divergent problem solving, where participants have to think of as many possible solutions as they can within a fixed period of time. So researchers went into this study with a hypothesis that acute stress will impair creative problem solving, particularly by reducing cognitive flexibility, and that these effects may differ between convergent and divergent tasks. Participants for this study included 70 healthy male participants with an average age of 19.24, and they were randomly assigned to one of two conditions, either the acute stress condition or the control condition. So to begin, all participants were given a demographics questionnaire to determine their positive or negative affect. This was known as PANAS. Then they were given an initial salivary cortisol sample, and then they completed either a stress task or a control task. In the stress task, it was the Trier social stress test. So participants had to conduct a difficult presentation and challenging mental math on a cold audience. In the control task, all they had to do was talk without anyone in the room and conduct pretty easy mental math in front of no one. Then a second salivary sample was taken. So after this, both groups completed both the divergent and the convergent task, and these were counterbalanced to make up for order effects. So in the divergent task, they had to generate as many possible uses for 3 common household objects as they could, and this was scored for fluency, flexibility, and originality. In the convergent task, they just had to complete some word problems that had a point scoring system. After this, they were given a second affect test and a second salivary sample. Additional salivary samples and heart rate were measured in 10-minute intervals after this, and then there was a third affect test. So the first result is that participants had faster heart rates and higher cortisol levels in the stress group compared to the control group, which shows that the Trier stress test did indeed induce stress over the control group. In the convergent task, stress induction was associated with lower accuracy, but a faster response time. In the divergent task, stress was associated with reduced flexibility, meaning that there was less of a variety of unrelated answers. So the first figure I wanted to talk about is the time and salivary cortisol relationship. So all this shows essentially is that over time we can see that the stress group had a higher rate of salivary cortisol, as we can see here on the left, than the control group that remained fairly steady. So this shows that the Trier stress test worked. The second figure I wanted to look at from this study shows that there was only a significant difference in flexibility between the stress and the control groups. So here we have the 3 categories: fluency, flexibility, and originality, and then the scores here on the left. And we can see that the stress group and the control group, and this is the only one where we had a significant difference. So people that were under high stress and low stress had statistically equal levels of originality and fluency. In convergent problem solving, flexibility is shown in both timeline and accuracy. In divergent problem solving, because there are so many different solutions, flexibility is manifested in the variety of responses from participants. We saw that stress induced faster but less accurate responses in convergent problem solving and reduced the flexibility of solution production in divergent problem solving. The next study that I looked at was conducted by Nair et al. Also in 2020. So some background for this is that stress impairs creative flexibility creativity and flexible thinking, as we saw in the previous study. High levels of creativity have been shown to be associated with increased functional connectivity. This is really important for this study. We also think that a genetic susceptibility to stress can increase an individual's response to stress exposure. So I wanted to just explain this genetic susceptibility really quick. So there is a short allele here of a serotonin transporter that's associated with less serotonin reuptake. So we can see here that this certain type of gene have less transporters here than we can see here, and so less serotonin is being reuptaken into the neuron, and that makes people more susceptible to stress and depression. So in this study, researchers sought to answer how stress affects functional connectivity in language processing regions of the brain. They additionally explored how gender and the presence of this stress-susceptible short allele that we just looked at influence this effect. They hypothesized that functional connectivity between the inferior frontal gyrus, left medial frontal gyrus, left middle temporal gyrus, and the right superior temporal gyrus would be affected by stress. They thought that this effect would be greatest in individuals with at least one copy of that S allele, and that this effect would be greater in females. So their participants were 28 females and 17 males that were genotyped to determine whether or not they had that allele. So there were two separate sessions for this research. Participants underwent a version of the Montreal Imaging Stress Test or a no-stress control task. Both tasks were interleaved with task blocks where participants performed an association task that uses both convergent and divergent thinking. Then researchers examined the relationship between stress and performance on connectivity of language processing. They found that there was no main effect of stress on functional connectivity. Changes in the middle temporal gyrus with stress were correlated with more task solutions for individuals with the S allele, but not the L allele. There was no significant change in the mean number of problems that were solved correctly under stress and no stress, and there was no significant difference between the stress and control observed. They did find, that gender appears to impact the effect of the serotonin transporter gene on functional activation. Stress reactivity in individuals with the S allele may have a different impact on functional network integration in males compared to females. However, it's important that the researchers state that the three-way interaction should be interpreted with caution due to the small sample size in each subgroup. So we can see that here, that in the stress condition, there's a significant difference between males and females depending on if they have the S allele or the L allele. And then here on the left, we're looking at functional connectivity strength between the left inferior frontal gyrus and the left middle frontal gyrus. So this research suggests that neural responses to stress for processes involved in creative thinking can vary with the influence of genetics. The discovery of such differences might serve towards the development of biomarkers for enhanced cognitive susceptibility to stress. Overall, research from both studies suggests that stress impairs divergent problem solving and creativity. Genetics may influence how big of an impact stress has on an individual's problem solving. The size of the impact by genetics may differ between males and females. All right, thank you so much.